So hi everyone, I'm Dan Bernard, and I'm happy today to tell you about our work, which concerns the neuroendocrine control of reproduction. So when I'm talking about neuroendocrine control of reproduction, I'm talking about the role of the brain and the pituitary gland, which sits at the base of the brain, and the hormones they produce and secrete, and how those hormones travel through systemic circulation to regulate gonadal function, so the testicular and, as I've depicted here, ovarian function. So in response to these signals from the brain and the pituitary gland, the ovary will synthesize steroid hormones and other hormones which feed back to the brain and the pituitary to regulate their own production. Other things that these hormones do is govern important aspects of reproductive physiology, including the growth and maturation of the ovarian follicles from which the egg will eventually ovulate. So in particular, we're interested in a pituitary hormone called follicle-stimulating hormone, or FSH, and this is just a picture here showing the cells in the pituitary gland called gonadotropes that make FSH. And the reason that we're interested in this hormone is that it's an important hormone. If you don't have it, you're infertile. So this is particularly true in women. It's also a clinically relevant hormone. And by that I mean in the infertility clinic, FSH itself is often given to stimulate ovarian follicle development prior to in vitro fertilization. The FSH hormone is dimeric. It has two subunits, and we study in particular the regulation of its beta subunit, depicted here in green. So I don't have time to go through all of the details, but FSH in the pituitary, its synthesis and secretion is controlled in a positive way by factors from the brain, like gonadotropin-releasing hormone, from factors within the pituitary, such as the activins, which positively stimulate FSH production and secretion, and then negatively by hormones that come from the ovary and the testis, which feed back to either selectively suppress FSH or do so indirectly by suppressing GnRH secretion. So what I want to do just very briefly today is describe our work on how these activins regulate FSH production. So for the most part, we use a combination of in vitro and in vivo models, and using an in vitro system, we worked out this detailed signal transduction mechanism through which activins, through these receptors and signaling proteins, actually generate protein complexes that sit on the gene that regulate the FSH beta subunit. And based on the in vitro model, we then use in vivo mouse genetics to test the predictions of this model. So for example, we may knock out different components of this system, which I have depicted on the next slide. So here we knocked out two of these components called SMAD3 and 4, and you can see in control animals we have nice expression of the FSH hormone in the pituitary, but when we knock out these genes, you can see that the FSH hormone selectively disappears, whereas the related hormone, luteinizing hormone, remains. And if we look in systemic circulation, FSH levels are undetectable, and as a result, these females are sterile. So we're hoping that through our work on FSH, we're going to help identify causes of infertility that are currently misunderstood or not fully understood, as well as potentially develop novel targets for infertility treatment. Thank you.